So hello and a very warm welcome to everybody here in the studio in Wageningen. Welcome everybody. I hope this will be a very interesting meeting, an interesting event, and we're going to talk about a very interesting subject already. Also, people that are watching at home, in their laboratories, maybe at their workplace, a warm welcome also to you. We have an interesting program for you this afternoon. We will start with a panel discussion, and hopefully this discussion will bring you new insights and views concerning the subject at hand. And that subject at hand is DNA analysis approaches in the breeding of asexually reproduced crops. As I told you earlier, we will start with a panel discussion, and we have experts from the plant breeding industry, and after that we will learn from breeders what their experiences are, what their lessons they have learned in the past, and what their views are on the future regarding the use of DNA analysis approaches. If you have any questions, please ask them. We have two ways to do that. You can ask them for the people at home during the sessions through our chat. And of course, the people here in the audience, you can ask your question by just raising your finger. And then Herco will take the microphone to you, and then you will ask your questions. The moderator for the questions will be Eric. Eric will be behind the scenes to see what kind of questions there will be on the chat. I don't know if we have a contact with Eric at this moment.
Yes, I'm here.
We only hear his voice, which is really nice because there's like it's like a voice from the sky. So Eric, are you ready for the questions that are coming in?
Yes, please, as many as possible.
Good. Okay. So with no further ado, let's start with the panel discussion. And before we start with the discussion, of course, we have to introduce our panel members. And our first guest is joining us from the United States. He is joining us from Oregon, Matthew Kramer. He is the former director of product development and commercialization at Fall Creek Farm and Nursery in Fall Creek, Oregon. He has more than 35 years of experience in the global research and management in plant breeding and in product development. And he has also numerous positions in both industry and government advisory boards involving plant breeding. So that number is impressive. Welcome, Matthew. You can hear me?
Yes, I can.
Perfect.
Can you hear me?
I can hear you perfectly. Thank you, Matthew. So first, a short question just to introduce you a little bit. You worked for the world's leader in production of blueberries and all its varieties. Is that a sector that could profit from the efficiency that DNA analysis approaches can bring?
Yes, we actually feel very strongly, or had felt when I was there very strongly, that there are a number of opportunities, maybe not so much to speed up the product development process, but to really stack the deck in our favor of successful outcomes. When it comes to blueberry breeding, it's a numbers game like all other crops. And indeed, large populations are required to get to the phenotypic outcomes that we're looking for. Being able to actually make smarter combinations based on knowledge around genetic diversity, as well as gene trait associations and the associated marker selection marker selected breeding work that we were able to accomplish was very actually helpful in not only controlling population sizes, but what we'd like to say, stacking the deck in our favor of successful outcomes.
Yeah, so I'm very curious to hear more from you. So I'm going to introduce our next guest, Graham Clarkson. You are the breeding director for Edward Vincent Limited, a company that already been growing fruit for 150 years. You have a very extremely successful breeding program over the last 25 years, and you are now implementing DNA analysis into your breeding programs. So Graham, welcome also to you. So what do you think? Is DNA analysis the logical next step in the breeding process?
Yes, thank you, Paul. I can only speak for myself and our business, but I'm here because it is and was the logical step for us that we've taken. We can see that, as has already been elucidated by Matthew, look for efficiencies, look to make better decisions, and to help us with doing traditional breeding more efficiently.
Yeah, I now already know that efficiency will be the magic word in this debate. So thank you. Our next guest, Chiopera, is an organization that represents breeders all over the world and especially helps them with the legal issues in IP protection, particularly plant breeders' rights. Joining us now is Dr. Edgar Krieger. He is the Secretary General of Chiopera. Welcome.
Thank you.
Are there any legal consequences concerning these techniques, these DNA analysis?
DNA analysis, I would say, I'm a lawyer, so my answer is it depends.
Yeah, of course.
Never give a clear answer to questions. No, I think in principle, no legal consequences for the analysis, right? Analysis is just you look at things, what happens, what you want to find in a new progeny or what you find in parental lines. The analysis is not the issue, but if you apply gene editing technologies, then there are some issues with plant breeders' rights and patents, but the analysis as such, of course, if you use a patented invention or a marker, then there might be an issue, but for breeding, for the process as such, I would say no.
Yeah, so there's no legal obstacles to implement these techniques?
No.
No, okay. Meili Tan, welcome also. You had an extensive career in the biotech and seed companies, and you founded your own company, Creanova Consultancy, in 2006. You specialize in the interface between science and business, and you give advice to plant breeding companies worldwide and to find the right technologies to accelerate their product development. So you acknowledge that DNA analysis to implement that in the process could give crop breeding a boost, but is it the only tool in the shed we can use?
I think that there are more tools for the breeder to use from and to help to accelerate the breeding. And I also think that the combination of DNA analysis with other disciplines like biochemistry, plant physiology, but also cell biology and plant tissue culture, that makes, I think, the strongest synergy and contribution.
Yeah, so DNA analysis is one of the tools. It can complement the other tools we can use to accelerate the breeding process. Yeah, good. So we have some statements prepared for you. So on the basis of those statements, we're going to debate a little bit, and of course, we have our guest in America who will be joining us also for this panel discussion. So, without further ado, let's start with the first remark, and that is for breeders, the benefits of using DNA analysis approaches can reach the benefits that are already achieved by breeders of crops like corn and rice. Who would like to comment on that statement first? Maybe Graham? Yeah?
Yeah, why not?
Yeah.
Again, repeating myself, which I'm in danger of already,
Yeah.
but I wouldn't be here and we wouldn't be doing it if we didn't hope to achieve those benefits.
Yeah.
We can see that the small increases that are being made in wheat and rice and those crops, those yields, are now being driven by the detail they're going into for the DNA. We're not as far on the journey in soft fruits, which is my case, but I do hope that we can start to grab more and more gain through using that analysis.
Yeah. What is the main gain from if you say crops like corn and rice? What are the benefits?
I think they've gained in terms of yield. I think they're gaining in terms of disease resistance and salinity, but they're not my crops. I'm speaking on what I hear and read and hope.
Yeah.
As a consumer of those crops.
Yeah.
Or user.
Matthew, what are your views on this subject?
So I would agree. I would say that there are real opportunities for improvement. We're way back in the early stages, I believe, of what's possible in these crops. It's really about information. So we're really, I think, in an information gathering mode currently, and that's where the DNA analysis is so exciting and so useful. Not only the gene trait association work that's being done, but also the understanding of how to deal with polyploids. Many of these crops are polyploid in nature, which makes the analysis more difficult. But I do see big opportunities ahead for things like yield, as has been mentioned, but also fruit quality in the case of berries.
Fruit quality. Well, berries, that's your field of expertise, soft fruits. Yeah. Quality, magic word?
Yes, quality is what we're looking for. Firmness, flavor, shelf life.
Yeah.
Those things that add on to yield.
Yeah. Miley, can you elaborate on that?
Yeah, I think if you look at the vegetatively propagated crops, the complexity of those crops are probably more than in rice and in corn, because they're polyploids. They're also sometimes difficult to cross. And there are so many different bottlenecks that we still have to overcome before we can apply, let's say, all the technologies that we have developed for the corn and the rice. But I think we are now at the right time in trying to apply that what we know, the knowledge and the technologies for the vegetables and the cereals for these kind of crops, to make it more visible what we are doing, more targeted, and also have more efficiency in selecting the right parental lines, the right hybrids, and also to follow them in the inheritance of these complex traits, especially those complex traits.
Yeah. If I can ask you, you mentioned bottlenecks. Can you mention those bottlenecks? What are the more important bottlenecks before implementing these techniques?
If I look at the whole, well, not only the soft fruits, but also the trees, the shrubs, and other tropical crops, tea, coffee, and so on, you see that in those asexually propagated crops, you have a very broad heterogeneity. And also, it's very difficult to trace back where the varieties are coming from, because they have been selecting all those years. There's no real breeding except for breeding at institutes. So that means you have to identify first what kind of germplasm you have. So you have to start very, very basic. And then we have the difficulties in flowering time. Flower fertility is sometimes inhibited. So there are many, many different things to overcome in your breeding program. And I think per crop you have to look at what are the bottlenecks and then find the right technology that's appropriate for that.
Edgar?
Yeah, and I think there's no alternative to DNA analysis. We need it. We have climate change. That is what we see. So if we want to make progress in these things, in climate change, to work against droughts or water shortage is an issue. We see extreme weather conditions, frost in Brazil, which kills the whole table grape industry almost in Brazil if it is too tough. We have labor shortage, we have energy prices going up, so we need to try to adapt the plants and the varieties to this situations. And there is no alternative.
No, that's interesting, because we see a lot of trouble in the world again with climate, with workforce. So we need DNA analysis to overcome those problems. What do you think?
Yeah, DNA analysis will complement and help us find what we're looking for.
Yeah.
And I think what maybe can add to this, we are far behind agriculture and vegetables. So fruit may be a little bit more advanced. In my feeling, ornamentals, there is still a way to go. Because we have almost 2,000 species, different species. In agriculture, you have 20, 25, the big ones, which will bring the money. And in ornamentals, you have 2,000. So then you need the markers for 2,000 and instead of for rice. So that is an issue also. And the structure of our companies is small, medium-sized. If you see the agriculturals, all of them are big, many of them are big, so other financial means. So that's a challenge.
Yeah. Do you see? You represent a lot of companies. Do you see them embracing these techniques?
Yeah.
Are trying to embrace these techniques?
Yeah. We made a survey as a short preparation of this webinar here. We made a survey and asked who is using smart breeding technologies, precision breeding, genetic engineering, conventional breeding. So all of them do conventional breeding, of course, as the basis. The ornamentals doing smart breeding, so that means DNA analysis for marker-assisted selection, precision breeding, that means so gene editing technologies, genetic engineering also, ornamentals more than fruits, because nobody wants to eat a GMO apple at the time being, because that's okay. And what we also see, and this is concerning, outside the EU, the breeders are more working on these modern techniques than inside the EU, which shows that the policy in the EU is not in the right direction.
Has that something to do with legislation?
Definitely, yes.
Yeah?
If you see the European Court of Justice decisions on what is gene editing technologies, they say it's like GMO and have to be regulated like GMO, which is a mistake, and it is unique in the world, more or less. That hampers and limits the possibilities of breeders.
Yeah. So outside the European Union, they are more lenient?
More liberal, yeah.
More liberal.
Liberal.
That's
Yeah.
maybe the correct
It's
word,
a
yes.
different approach.
Yeah. Marlie, you advise companies also about this subject?
Yeah, it really, again, depends on the crop and the problem they have in the breeding program. Of course, not all technologies can be applied for a small crop because, of course, budget reasons. But also there are certain technologies that are, let's say, less advanced, but still can help the breeder to overcome certain crossing barriers or even reducing the chromosome numbers at a stage where you still can do the crossings. So the fertility is still there to continue your breeding. So I think with the current toolbox and also with certain, let's say, combinations, so interactions between disciplines, so not only the molecular biology, not only the DNA analysis, but also other tools, other disciplines like the cell technologies at the moment that are being used to enhance and also to help the breeders to come to breakthroughs in the breeding.
Yeah. So, Matthew, a few minutes earlier we talked about that there's a difference between approach outside the European Union or inside the European Union. You are outside, you are in the United States. So what's your view on this?
Well, my view really has been colored by the fact that all the programs I've been involved in the past have been global in nature. And so we've always had to respect the local sentiments, the local rules.
Mm-hmm.
It is less restrictive outside the EU. That probably does hamper innovation in that geography. But nonetheless, many of these companies have found a way around that by placing some of this research outside of the EU.
Yeah. That's how you bypass.
Well, I don't call it necessarily bypass. That sounds too devious.
Yeah, it has a little bit of a negative connotation, I understand.
Yeah, it's really more of being opportunistic, right?
Yeah.
But, as far as I'm aware, these organizations, these research organizations around the world are very sensitive to public opinion and really do want to follow the rules. At the same time, they want to innovate, and so they design their programs accordingly.
Yeah.
One thing I would say about DNA analysis and breeding programs is that breeders are very smart and very creative people. And my experience has been that the more information they have, the better the decisions are that they make in terms of where they're taking their programs against certain targets. Things that can be done that are really quite simple in a lot of these crops could be something as easy as a genetic diversity analysis of your germplasm pool, just to see about, are we just remixing the same genetics, or are we actually bringing variation in via this program? So there's big opportunities for improvement in a lot of these horticultural crops early on, using some pretty simple techniques that really aren't going to run afoul of any kind of either local or national regulation.
Yeah. Okay, thank you. Before we go to our next statement, let's check in with Eric if there's a question online, or maybe there's a question here in the audience. First, Eric, do you have a question online?
Yes, Paul, we have. We have a question from Nicola, and it deals with, say, the European Court of Justice and what is said. The question is, are DNA analysis technologies fully accepted by the court?
I think this is a question for you, Edgar.
It depends again.
Yeah.
Depends on the court. And depends on who is doing the DNA analysis. So there are accredited labs, and usually these analyses which are done by these accredited labs are accepted by the courts. On the other side, they give, especially if you speak about an infringement in plant breeders' rights, it's still phenotypic. Comparison is the final proof, but more and more courts accept DNA analysis as either a strong indication or even a proof, which is much quicker than phenotypic comparison. So the answer is depends, but more and more courts accept this, which is also needed.
Yeah. Okay, before we go to our next statement, is there a question in the audience? I see a question over there.
Thank you.
Please state your name if possible.
My name is Anko Sørensen from Kijin.
Yeah.
I have a question for Mai Lee. You said a couple of times that you would like to see synergies between different technological fields. As an example, DNA analysis and, for instance, cell biology. Could you give an examples where that has worked very well? And if not, could you give us your best dream of what you would like to see happening in that area?
Okay. I think in the past, we've seen a lot of a combination of DNA analysis being used in the combination with cell technology, cell biology, especially for the crops that we are talking about, asexual propagated crops. You see that there are possibilities to overcome crossing barriers in certain crops, like citrus, for instance, where it's very difficult to make crossings, and where they use, for instance, protoplast fusion technology. And in the past, it was like trial and error. Of course, you cannot predict what you will get out of that. And with DNA analysis, you could pick the parental lines that you want to have and make the combinations that you like to have, and also analyze at the end what product you have. And I think without the DNA analysis, in this case, it would have been trial and error and making a lot of somatic hybrids or hybrids. And I think with the technology that is now also being developed for cell analysis and also cell imaging, we can have a lot more synergy with the combination of DNA analysis, where we can identify the cells which we would like to get and to select, and pick them and culture them individually. You can also do single cell analysis on that cell and try to regenerate from that cell back to the plant again.
So we have another question online, so we go to Eric.
Thank you, Paul.
Yes.
There's a question from Hanne, and there's a combination with Patricio. So it's about the packages. What kind of packages of DNA analysis tools do we foresee that will help breeders to make breeding time shorter and more efficient, like explained with the others?
Which one of the experts would like to respond to that question?
Yes. We can't look at Matthew. He's there.
Yeah.
I think Matthew's already said it. We are very much at an information gathering stage. It's very early in my business's adoption of DNA analysis. So we are collecting. We're probably doing things that rice and corn breeders that we spoke about would have been doing 20, 30, 40 years ago. You know, I'm looking for QTLs. That's early.
Yeah. All right, so we go to our next statement, which is an interesting one, because we already talked a little bit about this statement, and it's about new employees. So the statement goes as follows: breeders of asexually reproduced crops need to hire new employees that have expertise in this field. We already mentioned that at this moment in the Netherlands, but I think all over Europe, it's very difficult to get new staff, experienced staff. Do you agree with this? That it's difficult?
Recruitment is always difficult.
Yeah.
To get the right people with the right set of skills. is a challenge. So for DNA analysis type work that we're doing at Edward Vincent Limited, we have been out and recruited people with academic backgrounds in those areas, and then we try and bring them into being plant breeders. We're also taking on graduates to put them through our processes and our schemes to skill them up, and we train as we go. We're learning every day. Something like this is part of my training. It's all new.
Yeah. Matthew, United States, how is the situation there concerning
So
well-educated?
as has been said, recruiting is always a challenge, okay? But, in the US, we have quite a good pool of candidates to draw from. I'm a big believer in, indeed, we need to expand the skill set in the breeding programs in these asexually propagated crops beyond what exists today in most of our small organizations around the country. We're lucky enough that with the land-grant university system and some of the public funding that's available, training has not so much been the issue in the DNA-based techniques. Where we're struggling is finding plant breeders. The partnership between these disciplines is really what's important, and developing and managing those partnerships is what's going to lead to success. So yes, to answer your question, these people need to be hired. I don't particularly think it's a good idea to turn them into plant breeders. I think it's the best approach is really creating a partnership between our breeders and our bioinformaticians, so that it's an integrated program that hopefully is more efficient and leads to better outcomes. Speed, we're not to the point, I think, yet where things are speeding up, but more positive outcomes are what I'm helping people look for at this point.
Yeah. Miley, I see that you agree with what Matthew was saying.
Yeah, I think the best team is actually an interdisciplinary team.
Yeah.
Working together from different disciplines and also trying to understand each other, because they usually speak their own language, a plant breeder or molecular biologist, and I think that should come together if you have an interdisciplinary team of people that could work together.
Yeah. So we have a question online, and it's about artificial intelligence. So Eric, are you there?
Thank you, Paul. There's a question from, sorry, hang on. Wrong show. Sunil. Sunil is asking, how do we see the future of artificial intelligence and machine learning in DNA analysis? Can it really help? Can it speed it up? Or are special thoughts about it?
That's a big question, I think. Matthew, maybe you can answer this question, or
Maybe not.
maybe not. All right.
Yeah.
Maybe Miley?
Yeah, I think one of the exciting things is that at this moment there are a couple of developments in AI, and I think the opportunities are there to design machines that can help us to select cells, to identify cells automatically, and also it will take away the trial and error thing, because sometimes people can recognize, identify the cells that can regenerate well, but of course that is something that we can develop also with AI and automatization.
Yeah. I really want to go to our last statement. We have a couple of minutes left, but I think we can squeeze this last statement in, because it's such a nice one, I think. Because we are talking about DNA analysis, we are talking about artificial intelligence, but how important is it to have green fingers? I don't know if it's also in English the correct word, but green fingers. In German, we call it Fingerspitzengefühl.
Yeah.
Intuition, to know exactly what you are doing. Matthew?
Yes, I think it's very important, and that speaks to really the partnership that Mei Li was alluding to. The interdisciplinary team is what's going to make this a success, and green fingers, as you call it, is essential. There's no question about it in my mind.
Edgar?
I fully agree. When I speak to experienced breeders, I'm always impressed on their knowledge and how to select parental lines and parents, and how to screen, and just with their eye, they don't need a lot of machines, Of course, machines help and DNA analysis helps, but experience is a big value if you have it in breeding.
Yeah, I
course,
completely agree as well.
You work for a company that already is doing this for 150 years, so yeah. Yeah.
Yep. DNA will add and give us further tools, but you'll still need the breeder to go and touch and look and select the plant at the moment. Yeah.
So Miley, maybe this is also like a threshold for implementing DNA analysis, because maybe breeders trust on their instincts a little bit too much and they are not eager to switch to the more scientific approach? I don't know if this is the case, but...
Of course, their experience is important because they see the plant every day, every week, and it's difficult to teach somebody else what you see and what you feel. Mm-hmm. But I think anything that can help, and of course DNA analysis can help with that, also the new technologies also, but the feeling for the plant, whether it's in the field or in the lab, I think that's something that is important.
Yeah, and of course it's one of the tools we mentioned earlier. Also the touch, the experience of the breeder.
Yeah, and breeders are very smart people, and they know their limits. So if they see there is a tool which can help them to speed up or to have findings which they wouldn't have otherwise, they go for it. Some a little bit quicker, some a little bit slower, but they will go in the direction which is needed.
Are breeders very conservative? Maybe?
Yeah.
Yeah? Maybe too conservative?
No. Just
no.
right.
No.
Matthew, no?
I mean, it depends, as Edgar likes.
That's a very legal answer, as we have learned from Edgar.
But I do want to make one comment here, if I may.
Of course.
You're right that historically many of these breeding decisions were made based on intuition, but I think we've reached a tipping point in today's world. I think that the consumers are demanding higher quality products, and retailers who supply those consumers are demanding those products every day of the year. So the way I like to distill this down for the teams that I work with is pretty simple. Intuition should drive testing. Data should drive decisions. And I think if we can follow that axiom in our programs, we're going to make some real progress.
That's a very, very valuable lesson learned from you, Matthew. Thank you very much for this. So are there any questions in the audience or maybe online? Let's check in with Eric. Any questions? Yes.
Yeah, Paul?
Yeah.
We still have a question from online, indeed. And there's a question about Europe probably or possibly lagging behind. Johan is saying that in the United States or in Americas, plant breeders are well trained in using DNA technology, and he feels that Europe is lagging behind. Is that the case? And if so, how did it come so?
Who can I ask this question? You are looking a little bit... Maybe Miley?
I'm just thinking...
Are we lagging behind?
I don't think so. I don't think that we are lagging behind, but what is most important is to get support, financial support, if you want to apply those technologies in a small company. And I'm not sure if that is something that being more, let's say, supported in the US. And especially for the small companies, the hurdle is sometimes big because they think, okay, we don't have the budget for it, and they have to run their own breeding program, and then beside it, you also have to have the money for the new technologies. Mm-hmm. And I've seen in a lot of companies that they had to separate the budgets. So you have a budget for plant breeding, and you have a budget for new technologies. Otherwise, it will compete always, and it will not get the worth for the new technologies.
Yeah, yeah. We had a conversation earlier, and you told me that because it takes such a long time before you get the benefits from DNA analysis, that you have to be an early adapter to be successful?
Yeah, well, I think You have to
So
be on,
no lagging behind.
you have to be on the bus
Yeah,
to be able to do it. Yeah.
Yeah. So how did that go with Edward Vincent?
Very good question, Paul.
Thank you.
Don't know if we've got time for this.
We'll have a couple of minutes to spare. Yes.
But for us, it was the next stage to go to. We wanted to keep breeding well, traditionally breeding and getting better and better and better. And we felt we'd had to adopt these technologies, but we had to allocate funding and allocate timelines and justify it. In a business that is a fruit farmer, a plant propagator, and a plant breeder, if I had 100,000 euros, pounds, dollars to spend, it's all the same now.
That's true.
Do I buy 2 new tractors and make a return next week, next year?
Yeah.
Do I improve my propagation systems and get a return back in 3 years? If I invest in DNA technologies, I hopefully will get more efficiencies and better chances of breeding better in 10, 12, 15 years' time. That's where you have to make a decision.
So it's an investment for the long run. So it's also, well, not a risky investment, but you have to think about it.
Yeah, you have to think about it, and the business has to want to do it and to understand the benefit that it's going to get there.
Yeah. So do you think a lot of your fellow breeders, they go for the new tractor instead of the DNA analysis?
I'd always want to go for the DNA analysis.
Yeah.
We need tractors as well.
Yeah, of course. Yeah.
Both, huh?
Yeah.
And what we see, especially in the last years, DNA analysis, genetic engineering, gene editing goes really steep up, and there's, as I said, there's no alternative. I think the bottleneck, as Matt says, it's the traditional plant breeders, the new generation, because the universities don't educate plant breeders. Not everybody is in a situation to have Wageningen University.
Yeah.
I think there are many young people who are going for biotechnology, so that I think here the people are there, the knowledge is there, but in Europe the hands are bound when it comes to the application and the commercialization. That is, of course, also a thing.
Yeah. So this is all we have time for during this panel discussion. It went very fast. Thank you so much for your contribution. Also, Matthew, thank you very much from the United States. You got out of bed very early, so again, thank you very much, and hopefully we will see you soon and hear from you soon. Thank you. So we go to our demonstrations, but first let me invite you to have a seat in the. Oh, before you go, is there any question? Any question we can ask the members of the panel at this moment? No? Very good. Thank you. You were very complete in your answers. So we are going. Thank you. Yes. Yeah. So now we are going to hear from a few experts from the field, some breeders that can tell us a little bit about their experiences in the past and also the lessons they have learned and also their view in the future of what DNA analysis can bring. So first I want to introduce Dr. Dominic Lawsuit. He is the director of breeding pot plants at Selecta One, and for some years now, you can join us. And for some years now, in addition to his own garden, he has been professionally involved with the attractiveness of plants and their ornamental values. So Selecta One is a company that uses different experimental methods in the breeding programs, and Dominic Lawsuit is here to tell us a little bit about those experiences. So, Dominic, you have some water. I would say the floor is yours.
I start with this.
Okay, yes.
So, well, many thanks for the introduction, and I'm really happy to be here and present some of the aspects here. At the beginning, I would say we are at the same level than we heard before, more or less. We are lacking behind of the agriculture decades, I would say. But we are gaining out of their experience, what they find out, and so on. And I just start now with my presentation. And first is just one slide about us, that you know who we are. We are a company. Our main facility is located in Stuttgart, in Germany, in the car city, I would say. So we heard before about struggling with hiring good employees. We have the luck that we have an university nearby, the University of Hohenheim, where many of our employees coming from also. And they are well-trained, yes, but sometimes they're not in that detailed trained by genetic analysis. They have more these theoretical aspects, but the practical use quite often is missing. So just one short overview of our companies. We are spread more or less over the world, but I would go more in detail about the breeding department where I'm located. Our main facility, as I said, is in Stuttgart. We have additionally breeding sites in Italy, but also in France, for example, and with some partners in the US and so on. When I started at Selecta, I was a little bit surprised about how this business acts, I would say, because they really have this, you called it green fingers. I would say it more, the breeding was related more to this breeder's eye in this direction. The use of experimental designs, for example, was really limited. And now the last couple of years, I think it's changed in this business. More and more this expertise coming mainly from the agriculture, but also from other researchers and so on, are coming into our company or into our business. So just to have one or two pictures, the presentation here or my work in general is really colorful, and I just want to mention one or 2 points. We are dealing on commercial side with around about 80-85 plants. So we are working really broad, I would say. And with most of our crops, we cannot jump into this deep in the, that we do not like an agriculture company who just has a handful of crops. I just want to present 1 or 2 slides. These are, for example, Poinsettias, the Christmas tree, where we are really good in breeding in these aspects. In general, we are a traditional clonal breeder. We also use other techniques, but in general, I would say we are a clone breeder. Here are petunias, for example. This is one of our highlights, this sky petunia with these dots on the flower. And our last one by far, we are in this business, they called us the carnation breeder, because especially for the pot carnation, we are the world market leader. So then I jump over a short agenda. As I said, I just display here more a practical approach, not to go into this scientific area. And here is the agenda. At first, I explain or show you something about population structure, how to use it. This visualization tool, I would call it, then the marker as itself, what we can deal with the marker or how we deal with this, and then Jiva's approaches, and at the end, 1 or 2 examples. Just simple example, we do it more or less in the same way. I started with this population structure, and one approach to use this is a principal component analysis. I just display here an overview. It means more or less just shown the first 2 layers or the first 2 principal components. And you can see here out a nice overview over the genetics we analyze. In this case, one crop which you can see by the PC1 and PC2, the numbers there, the diversity is more or less low. This is a crop just breeding the last 20 years, starting with 3 wild types. None knows exactly which one, but there is a bottleneck at the beginning, and it's displayed here also by this percentage, which displaying only the first 2 principal components. What we can make here to visualize this a little bit is to check the quality of the genotypes, for example. I just labeled here in 2 colors. 1, 2 samples. It means I take the same genotype and check it. And you see here the green labeled dot. You just see 1. Even if this are from two different plants, but the same genotype. And then on the other hand, you see the red dotted, there is a small shift in between. I would say it was an external from another company, a genetic, and we received them once, 7-8 years ago, and then just 1 or 2 years ago, and we compare it. And you see there a slightly shift. It's maybe a kind of selection internally from them. This you can visualize with this kind of tools. You also can see how the diversity acts if you make different crosses, in this case 3 crosses, and you can here visualize really easy how narrow they are or how broad they are if you use one crossing. So you also can compare, for example, with your competition. Here I just labeled the competition varieties I used for this analysis in red color, and you also can go into detail. I mentioned these 2 companies here. One company is the biggest fish, I would say, in my business is the Min Orange, really huge company. And in this crop, they have more or less the same strategy than we have. They use all what is available of the gene pool. On the other hand, you see the next company, Sakata. They focused mainly on one topic or on their own genetic, and it's displayed here also in this population structure. So this kind of visualization, I would call it, you can use it in different forms. Here also you see another crop, and what I want to push that you have a look to this is the percentage of the principal component here. It's a much more diverse crop in this case. The diversity is much higher, and you see here a clear structure. The colored dots represent the genotypes, but it's separated here by the concept of pool building, I would say. You see on the left-hand side, these green-colored ones is more or less one pool. On the right-hand side, the lilac ones, and in the middle, more or less in this case, it's just separated by one SNP marker. You see a clear differentiation, and in the middle, directly the heterozygous form. So same you can do in another way, that you have here a gene pool, in this case, our cut carnation, one of our crops, and I just labeled the single genotypes in their longevity. It means the vase life, how many days they survive, the cutted parts of the plants in one vase. And you've seen it here, if they have if they are green labeled, they survive longer. If they are red labeled, they died quick. Yeah, you can use this kind of analysis here. Also the phenotypic data, it's a nice distribution here, and the heritability is quite high. We use this approach for 370 genotypes in this case. I think these are good data you can use for a genetic analysis. This was just one part. All of you know this phylogenetic trees, for example. You can also build this out of without any phenotypic data by the genetic marker data you gained. Then the marker is itself. If you have a reference card, this is the best approach. In most cases, in my business, we didn't have. I said we're dealing with so many different crops that it always depends on how important is this crop for us, how much money we spend on this crop, on a single crop, and the reference card is still not cheap, I would say. But here we have it. One I show here, or I display in this case, it's after a kind of quality check that we have gained 30,000 markers for this crop, distributed over the 12 chromosome. And with this violin plots, so they called, you see where they're located. As huger there are more markers located on this location, on this part of the chromosome. Simple approach, how to use it. Our main driver is innovation, I would say, in my kind of business, to bring some new plant patterns, that they look in a different way, that you've seen something new, never was on the market before. This is really driven. And from my point of view, this has also something to do with the diversity. And a really simple approach to have an access to the diversity of the markers is the so-called PIC value, the polymorphism information content. Here is just the formula, but to make it in a much quicker form, this PIC value can be between 0 and 0.5. And if you are more on the side of 0, you have a kind of a fixation of one allele on this point, on this marker. And if you have it more on the side of 0.5, then you have more diversity. And how you can use this, we heard this before in the discussion here in front. For example, more and more, not only this phenotypic description between the profiling of a genotype, you can use also this marker-driven ones. And a simple approach is just to pick out some markers with a high PIC value who shown a huge diversity and distribute it all over these chromosomes or over this parts. It's a really simple approach, quick done. You do not need any phenotypic data. You just need genetic data. Then the next point on my agenda is the so-called Jivas. We use this approach for our most important crops now. And as I said before, and we heard it also before, we are lagging behind agriculture. But for us, this simple Jivas approach are really useful. In most of our crops, nobody do some genetic analysis before. And quite often, we find huge parts explained, major QTLs, and so on. What is really important, and I think this is what also was discussed here before, is this combination of different tools, I would say, because one major aspect from my point of view is also this evaluating of the phenotypic data. This goes into the direction of green fingers or of this breeder's eye. I like this terminus more, I would say, but also to structure your kind of field trials or greenhouse trial with experimental design to avoid the residuals or to avoid some influence of shadow and so on. Yeah, just mentioned here how we build it, how we calculate our phenotypic data. It's more or less influenced the phenotype, what you've seen, what kind of plant by different points, by the genetic as itself, by the environment where it grows, by some aspects like pinching. This is especially related to us, for example, not for other businesses, but also by the use of plant growth regulators, by where the cuttings coming from. It's also influenced strongly the phenotypic act, especially in the early traits, like how early they bloom. Then some where they're located inside of the greenhouse or in outdoor fields, and the year, and by all the interaction between all of them. So it's just one slide, short done, general introduction into Jivas. What I like most by Jivas, it's the first point here on this list is hypothesis-free method. It's just needed that you have phenotypic data, good phenotypic data and the genetic data, and then you can start. And this is for me one of the best points in this kind of using Jivas. So in general, it identifies points on the DNA who are associated with some phenotypic traits. Here, just a simple approach. I display the two alleles here, just color-wise. It's not scientific approach, but to visualize it, here the red one, the red allele, let me call it this way, stand for a filled flower, and the blue one for a single one. Yeah, in best cases, you find something like that you have on one side, you see here a so-called Manhattan plot. It means if this marker, in this case, these are 32,000 markers, we know the exact position over the chromosome, and I didn't bring a picture in. This is, I find a major QTL, or better, you can call it not a quantitative, it's a qualitative trait. In this case, it's explained 87% of the variation here. It's a really good marker. You can use it directly, and it's related to the position of the flower, if it stands upright or is hanging. Yeah, then more or less at the end of my short talk here, I pick some example. One example, this crop is called Calibrajoa. Calibrajoa is a huge crop in our ornamental business, mainly driven by the US market. There is it by far the hugest crop, and in general, it's called now the hugest ornamental in the world, more or less. If it's the true, I don't know exactly, but it's a huge crop also for us. And you see here the different grades of filling. On the bottom, you see a single one, and the others have different degrees of filling, so it means the reconstruction of the petaloid in this case. Here you can see some box plots where you see on the x-axis the two different alleles, and on the y-axis the level of filling. If it's more on the bottom, on the y-axis, it means a single one, like the one here on the bottom, this yellow with red veins. And if it's more on the top, a really good filled ones. And you see here a clear distinction between these two, and we do at the beginning a sequence-based genotyping approach. This is labeled here in, what is the color? Blue color. And afterwards, we verify it with some cast markers, a really simple approach. The cast markers are quite useful for breeders, I would say. Yeah, and it's quite good verified here. And what I realized by analyzing this was there were just the homozygous alleles or the homozygous forms available. And for me, it gives a big hint that it's located on the mitochondrial DNA. We have a reference of the mitochondrial in this case, and we can simply blast it and know exactly where this marker is located on the mitochondrial DNA. Then I come to my last example. You've seen here a picture of the phenotype. Should reflect the vigor. We rated it from 1 to 9. It means on the left top, this plant is 1. On the right-hand side, on the bottom, is 9. Simple approach. And we do the same kind of analysis and verified it afterwards, and we find a dwarf gene. It was known before, but we now combined these two markers I shown here for Calibrajoa, this compact trait and this trait of filling, to have a patent in the US. It's now granted of the combination of these two traits or these two markers in this way. So, and this was it. Many thanks.
Thank you, Dominic. So we have time to ask Dominic some questions. If there are any questions in the audience, I see a hand there. Wait 1 second for the microphone. Yes, on the third row. You have a question for Dominic.
So, hello, I'm Li Ping from Kijin. So my question is, do you consider, because today we talk a lot about combination of technologies, do you consider also implement digital phenotyping in breeding program?
We try this, and also we start this also together with external company. I would say they are experienced in this way, but at the moment we decided not to follow it in such a deep way. It's related on one hand by budget, on the other hand, really I, for example, make my PhD also with this sensor topic, and I realized there are some quite good sensors where you can reach much better data, much accurate data, but in general, for most of the traits, I would say, and especially the complex traits, the human eye is really good. My point of view.
Okay, thank you.
Thank you. Are there any more questions in the audience? I also would like to remind the people that are watching at home that you can also put your question down into the chat so we can relay those questions to one of our experts. Thank you, Dominic, for your presentation and for your insights.
Thank you, Paul.
And I would invite you to take your chair in the audience and listen to your colleagues, because we are going to our next presentation. And we are going, yes, of course. Thank you, Dominic. Because we are going to talk about a very important crops named potatoes. So the development of a new race of potatoes will take years. It will take up to 12 years, I think. And our next guest knows exactly how this process works and how the breeding process can benefit from using DNA analysis. I would like to invite Guus Heeselmans, who is already here. He is he's the research director for Maya Potatoes. So you can tell us, of course, a little bit more about your experience. So Guus, welcome. And the floor is yours. Thank you.
Thank you, Paul, for this introduction. Thanks, Kijin, for the invitation. I think this is a very good opportunity to share our experience, to learn all, to have a better progress with our work in DNA analysis. And my title of today is, Does DNA analysis boost potato breeding? With a question mark. I come to the answer at the end of the presentation, but before we go to that, I would like to tell you some more about our company and about our breeding program and how DNA analysis is implemented in our breeding program. Our company, this year we celebrated our 102nd anniversary as company. We are a family company in potatoes since day one. Nowadays we are a breeding company, but our turnover comes from the seed potatoes we handle, we have all over the world, but also very important is our potato grown on the license base. And that will also pay the breeding back, of course, and that makes it possible to have new evolutions in your breeding program. We are an international operating company. We have daughter companies in the UK, in Poland, in Turkey, and in Spain, and that are very important points for further work with our varieties. And besides that, we have a lot of agencies where we have the varieties traded on the license base. Yes. Our varieties, we have in the potato, we have 3 segments. We have crisp potatoes, fresh potatoes, and French fries. That are completely different markets and also different approaches in the breeding. So it's always important to have your plan ready when you are aiming for a variety in that specific group. Our oldest variety is Lady Rosetta. That variety brought us all over the world because it's a crisp variety. And the processed varieties, if you have a good variety, then the chance is very big that you can have it grown all over the world, if the variety is suitable for that, of course. Some history of our breeding program. We are a relatively young breeding company. In 1985, we started the breeding in Rilland, in the south of Holland. We started with 30,000 candidates each year, so 30,000 seeds, genetic different seeds to select out the best variety. And we had some steps in the history which were important for the development of our programs, which is shown in this slide. But very important is 2009, when we established our DNA laboratory. Thanks to a big program we joined in Wageningen here, the CBSG program. In that program, wild species were analyzed for resistance towards late blight. And a lot of wild species were analyzed, so we found back a lot of wild species who carried resistance. But on the other hand, we also got the gene, we got the marker. So we had a toolbox to do the first serious steps with DNA analysis at home. So we established a DNA lab, and before that we hired a fresh young breeder who had some more actual knowledge on DNA than the small team we had. And so we started with a small lab, and then you can see how it grew through the time. So we start with electrophoresis, then we had light cycles, and now in 2015 we have our third generation of DNA analysis techniques. Another very important decision-making for our future at that time was a big crisis we faced in 2014. We were having PSTVD in the heart of our breeding station. That was a real disaster because we had to throw away all our material, and we could keep a little bit of our parental lines. If not, yeah, we maybe would not have survived this. But on the other hand, that was also a boost, and then you see the value of a family company who can look forwards and also have a flexible approach. Together with the board of Meyer, we decided to really invest in new techniques and to speed up our program so that we could win back the loss and to be on the head of the peloton, as we call it in cycling terms, for the future as well. So that was a very important but very crisisful year. And that also brought us to new thinking in how to deal with new knowledge. And of course, we are still active in nice big projects, for instance, from Wageningen University. But also, we also take our own plan by bringing partners together, interdisciplinary, like Melly already indicated, to find new solutions for our breeding program. Our breeding goal, our company slogan is everybody deserves to enjoy food. That's quite ambitious, absolutely. You see the word enjoy, that's quality, because we like to eat potatoes, so please do so. You have seen the varieties which you can buy everywhere in the world. But of course, they need to be available as well. And knowing what the future will bring us with climate change, with stricter regulations on chemicals, on fertilizer, yeah, that's a big challenge for potato breeding, but I guess that's the same challenge we all have to deal with. Potato is a one-season crop. That means in many areas of the world, storage is also very important. And also storage is limited with some regulation. So yeah, there are a lot of traits which needs heavy attention to go forward. The principle of breeding, I think it's good to determine two disciplines in breeding. First of all is the combination, the quality of your parental material, and the combination gives the chance of success. But if you have done that, you are not there, because after that you need selection program to find the proper one. And both disciplines need all attention to be successful. And here you can see the first DNA figure, and then you see that both in the combining and in the selection, I will come back on that, DNA analysis can help a lot. Our breeding program, in an overview, is we start with a crossing program, mostly determined for commercial selection, to find commercial varieties out of it. But in some cases, we have to go back a few cycles in a pre-breeding scheme, because, for instance, if you have a new resistance gene from a wild species, you have to get rid of all the unwanted traits, and therefore a pre-breeding scheme is needed. But there you can win a lot of time if you have the markers belonging to the gene of interest, and then you can speed up that cycle to go as soon as possible to the commercial breeding program. The commercial program, it lasts something like 7 years to go from crossing towards introduction of a variety. So here we see already a win of a few years, as in your introduction, and that has to do with the boost we gave in 2014. I will come back on that. This is a screenshot of our combination design, the crossing design. So a little bit of fingerspitzen, but also a lot of data. On the left, you see the phenotypic data of the parental value on phenotypic traits. Green is good and red is not good. And on the right, it's a little bit blurred, but that's our marker information on the traits from which we also can direct with marker and analysis. So and then you see the history of the crossings, which is also important to take in account. But now our parental material is analyzed on a lot of markers, so we can also predict the genetic distance, which we think is also important to make choices in your crossing program to make heterosis possible as much as possible as we want. So that information we take in the decision model, yes or no, to make that specific crossing. So we use both the phenotypic support, that's the data, and on the red you see the genotypic support. And the rest is intuition. Our selection program, so if we have succeeded crosses, we have a lot of seed in our closet, and then we have to make choices which one we want to sow out. First we look to the markets of interest, and then we look to the specific characteristics which are important for those markets. There can be diseases, but it can also be climate adaptivity. Of course, it's all, but you have to make choices sometimes. And then we shift the seeds in 2 routes. On the left, you see the route 1, this is the phenotyping route. That means that first we look to the phenotype on the field, and afterwards we use markers on informative base. On the right, you see the genotyping route, which means that we first select with markers selective on specific traits, mostly resistant traits, and then the survivors may join in year 2 on the field. So both columns will be merged in year 2 together, and you can imagine we don't care from which route the variety will come, but we only take the best. So in year 7 we are in a variety stage, and from year 3 onwards we do a broad phenotyping on quality characteristics, also depending on the market, of course. And you see also a little sign that we do some genetic studies to find new markers. Whilst phenotyping, you can use that information to find markers. And on the other hand, we also do some population studies to do so, more directed. We do also broad phenotyping on physiological aspects, where potato is grown in many soil types and many climates, many potential problems, so that is the choices of your trial fields is important to make choose the right spots. And of course, potato is a very kind host for diseases, and you have to take in account, depending on the region where you go, the resistance level of your new candidates. In the last phase of the breeding, we test our candidate varieties throughout whole Europe, and we want to cover every climate zone as possible, to have a prediction on the climate adaptivity. And here you see the G times E, which is a very, very important interaction in potato and in many crops, of course. And you see with the DNA analysis, that's a real future challenge. We don't have grip on it yet. In 2017, we started our own project, interdisciplinary, with partners who gave support in many areas of the plant breeding. Kijin was involved as well. So you see on the bioinformatics, on the genetics, on the data sources, fine phenotyping by drones. And that all together gave us new tools in hand to expand our breeding activities. And on the right, you see the graph of the amount of data points of our DNA analysis. Genomics, of course, we have two bioinformaticians. We have always a lot of questions to those bioinformaticians to find to find the needles in the haystack, the DNA needles, or to build a haystack out of the DNA needles. But most of all, I think the least you can do for your bioinformatician team is to provide them with the best phenotypes. Now we are running in a project with the same approach, but now it's more oriented towards abiotic stress. We have a combined stress approach for salinity tolerance, drought tolerance, and a low fertilizer regime. Also here we combine disciplines to find, in the end, genes, which is of course not easy with those quantitative characteristics. Okay, let's finish up with the lessons learned. People, we heard that already in the panel, keep DNA stuff, which are specialists. You have to hire specialists to do special actions, but keep them as close as possible to the total program, and the other way around, of course. Have them involved in the green work as well. The lab, be prepared for the future step to take, because a lab is always changing, and try to find the bottlenecks and solve the bottlenecks and go to the next bottlenecks to really speed up further. The crop for potato, tetraprid, vegetatively propagated. We have now good experience with snake-based markers, which gave us good insight also in the allelic frequency, which is important also for the design of your crossing. Expansion, of course, we want to have more markers, which lead you to a trait, to the phenotype. And up to so far, we use array approach or sequence approach, which is more new for us to find new markers. Last but not least, the data. You need a solid database and a good data flow to catch the information out of the data at the moment you are ready for it. To conclude, the answer to the question where I started with, does DNA analysis give boost to potato breeding? Yes, absolutely. Absolutely. It's a valuable tool to speed up, to have early information, to stack genes, to have more genes of one trait together, especially with resistances, to have more locks on the door, and to enlarge chances, the statistics. If you can put more seed in your system, you have more chances that the genetics falls in a good way together. Not to forget also that you get also much new knowledge in your own R&D department if you have your own equipment, if you have your own lab. We also notice that it attracts young people for stages, practical periods, and also we are now more attractive for new people, but still it's very difficult to find them. Maybe not just as difficult as finding new varieties. Then for us, the implementation of using DNA techniques in the timeline, first we focused on a few very important traits from which we could use the markers to use them in a selective way in our breeding program. And that takes us to more, so we try to find new markers using genomics And again, phenotyping, very important. And now we are using a broad set of markers to support the crossing strategy, which is important because before you fixate your genetics, that's the last chance, is making the crossing. And now we are learning, and we are far away from this, but we are learning to come to a holistic approach to give some answers to the G times E interaction. But that's for the future. I think I can conclude now. Thank you very much for your attention.
Thank you, Guus. Yes. So, as I understand from Eric, there are no online questions for you. Willing the time, because of the time, we have to move on to our next presentation. But I would say afterwards, after this event, there is Guus will be here, I think, sure. to answer your questions if you have them, and then hopefully you can get all the information you would like. So, thank you, Guus.
Thank you.
Have a seat in the first row, of course. And we will move on to our next presentation. And the next presentation will be by Fernando Garcia and René Hofstede. Those are two, two scientists that spearheaded a number of projects here at Kijin in Wageningen. Thanks to his 17 years of experience in a breeding company and his academic background, René Hofstede can advise and guide partners in choosing from, as you put it, the candy store called approaches in DNA analysis. And then, of course, Fernando Garcia, he's currently leading the banana breeding program. Very important task. Also, we heard lately that the TR4 disease, which is a fungus, as is well known, is threatening the worldwide production of bananas. So please welcome them. And of course, we are very curious to your insights. The floor is yours.
Yeah, thanks. Okay, so I will start this duo presentation by explaining what Kijin is doing in the field of vegetatively propagated crops, because that might not be obvious to all of you. And after I've explained a bit on that, then Fernando will take over about more details. So Kijin is all about innovation, technology development, and developing new technologies for use in breeding. At the same time, we're also working on application of those technologies in actual breeding programs. And we've been doing that for 30 years, mostly on vegetable crops, field crops, and flowers. But we see more and more clients in the fruit field. So in fact, the past 5 to 10 years, we've gained in experience in the fruit crops, also working there, applying again those innovative technologies. And for us, that in the beginning it was a new field because obviously it's vegetatively propagated. A lot of crops suffer from long generation times. They're polyploid, and the companies that were giving presentations earlier also show that there's a transition going to technology-based breeding. So what do? Basically, we adapt and apply technologies to those crops, and we invent new technologies specific for those crops. And you can think of mapping in polyploid species or determining allele frequencies, mutation technology in vegetative crops. It's all new. We do that in several ways. So we can collaborate on basis of a menu, so to say. We have a list of projects. We can simply carry out a project of your choice, let's say a SNP mining project or a GWAS. But preferably, we go one step further, and we like to think along with you on how to use the different approaches in your breeding scheme. So in that case, we make roadmaps, staging several projects and indicating what your company should do and what Kijin should do. And you can recognize it in this table. The green bars are the activities for the breeding companies. The purple bars are the activities for Kijin. We even like to go one step further, and that's to think along on the breeding strategies you're using. So besides advising, you can use these 2 or 3 projects in this time frame, we also like to think along in breeding strategy. And this is an example of 3 options, basically. Follow conventional breeding, do conventional breeding with the use of markers, or go to mutation breeding. So that's a simple example. This is much more elaborate, where for a certain crop we thought of all combinations of all technologies that were available at that time. And well, as you can see, it's much more elaborate and it's clearly technology-based. It involves mutants, for instance, DH-inducing genes, gene editing, even a transition from going from a vegetative propagated crop to a generative crop. Well, thinking about that, we thought it might be nice if we also do our own breeding. So for those of you who don't know, we have our own breeding program at Kijin in banana. And that's a joint venture between Chiquita, Musa Radix, and Kijin, and in very close collaboration with the group of Gert Kema at the work. And what is our own strategy? We call it reconstructive breeding. Well, if you think about the two most important accessions in banana, it's Cavendish and Gros Michel. It's more or less known where they come from, which types originated, or from which type did they originate. So that allows reconstructive breeding. And, well, in short, we combine three pillars, so to say. First, genetic resources, of course, as the basis of any breeding program. Use genetic tools, which Fernando will show more about, but also the crop knowledge, you should not forget that. And it's like, how do I get a banana plant flowering in a greenhouse? How do I make crosses? How can I store my pollen, or how do I deal with seeds? So that's also an important part. And with that, I'd like to give Fernando the floor to talk more about the genetic tools.
Okay, thank you, René, for the good introduction. Indeed, we are going to talk about bananas and the breeding program of a tropical crop in the Netherlands, and at the same time, we are going to show you how the DNA technology boosts our program. So as you know, similar to other asexually propagated crops, bananas are no exception and are very difficult to improve. So, physical barriers, genetical barriers, polyploids are bottlenecks that are very difficult to handle in banana. I hear my echo is very confusing. Okay. As you know, we have only one type of banana in the market. Actually, 50% of the global banana production in the world relies on a few cultivars of the Cavendish, the triploid type of banana. It doesn't matter the origin, it doesn't matter the sticker you find in each banana are genetically identical. That makes that the system of production in banana is very fragile, especially now with old and new challenges from the biotic and abiotic factors put the banana in risk. So there is no doubt that black sigatoka and Fusarium wilt are the most important fungal diseases in banana, very limiting. As you can see here, black sigatoka affects the leaves of the banana, and this is controlled by the application, in many cases, weekly applications of fungicides, as you can see in the picture, which really hampers the cost of productions of banana, and you can imagine the impact in the environment. On the other hand, we have Fusarium wilt, the famous tropical RIS4, a devastating disease that is spreading around the globe. Just look at those numbers. In 2013, 5 countries reported this disease, and in the last couple of years, we reached a number of 20 countries. 20 countries. That's better. It's very annoying to listen to your own voice. It's never nice. Okay, so as you can see, also very devastating disease. And the problem with this one is that you cannot control it. The previous one, you can control it, cause a big impact, but in this case, it's not possible. There are no replacements, no control. But Cavendish is not the only banana that exists, as you know. Actually, literally, hundreds of bananas exist, but we are just married with one. And that's the challenge, because the industry and the consumers want only one type of banana, and that's the challenge. We need to make and reconstruct, as René mentioned, type of Cavendish banana. So there are more than 1,000 different types of banana, including diploid and triploid cultivars, some of them still fertile, a known number of human-made hybrids, and a very important group of wild types. The wild types, of course, in every breeding program are the the mine of gold, because those accessions always have a lot of nice and interesting traits, especially yield, as you can see, resistant, but the most important is that we can use them for breeding because these are fertile. We can still cross them. So what have we done at Kijin? First of all, our breeding program incorporated a core selection of that diversity, and as you can see, we are growing them under greenhouse conditions here. You can see the roof there. They are constantly flower, which enable us to make all the crosses that we want. But that doesn't make us different to other breeding programs. So for that, we have the phenotype. We have evaluated all those accessions for resistance to black sigatoka as well as tropical race 4. The ones that have the asterisks are resistant varieties. And for those who are not familiar with this kind of graph, just look at this as a family tree. So with the sequencing of each individual accession, we know more about the genes, the markers. We can develop different types of analysis, but the most important part for us is that allows us to make those groups of different types of bananas. And as Rene mentioned, we are doing something that's called the reconstructive breeding, and that's the most important part of this graph because it allows to identify the most potential ancestors of Cavendish, and thus allows to go back in the evolution of Cavendish and try to make a new Cavendish, but this time using resistant accessions. At the same time and simultaneously, we have learned and built more knowledge in terms of our crop. So we know how to grow it. It's a challenge to grow. It's a challenge in terms of space working in a greenhouse, but it's also lots of opportunities to play with different characteristics and different situations, different conditions, which allows, for example, to improve the quality of the pollen, being more efficient in the selection of the parents and the crosses, the production of seeds, the production of mapping population, which enable us to do genetic studies, and also to play with the ploidy. As you can see, we are also generating our own tetraploids in order to perform additional crosses. So at this point, I feel that we are repeating the same speech as the previous speakers, and that shows that how important it is to mix different type of data, different type of analysis, different disciplines, different ideas, different ambitions. So that enables us to make a better selection. And in this graph, I'm showing you how we select the best parents based on the genotype and also the phenotypic information. Selection of the best parents based on which ones are resistant or susceptible. And at the same time, this is a case of a study how we managed to make crosses, develop populations, clone those seeds in order to have enough individuals to study and to generate new data as the resistance or susceptibility to, in this case, tropical race 4. Again, thanks to the combination of these different disciplines, we are able to take this as a complete unit and analyze the data. In this case, I'm showing you in the last picture a context in which we found a region in the genome that might be associated with that specific trait, in this case, the resistance to tropical race 4, and also with technology that we have at Kijin, like Cropedia, for example, in which we combine all data generated at home here, but also public available data related with DNA. We can actually go deeper and pinpoint specific candidate genes. So we analyze the DNA in different ways. I just show you one. But at the same time, we are also analyzing the RNAs. So in this case, as you know, the Cavendish are resistant to the Fusarium race 1, but susceptible to the tropical race 4. And when I'm talking in this slide about RNA, I'm talking about genes that are activated or expressed during the process of infection of the plant, for example. So in the first graph, you can see the Cavendish being attacked after 30 minutes in contact with the disease. And in the right graph, you can see all those red dots are genes that are activated, let's call it like that, after 8 hours. That allows to do different type of analysis, but initially confirm which are the genes that are associated with the resistance, but at the same time, and why not also analyze which ones are associated with susceptibility. That's another different approach. So everything I show you so far is part of a combination of pre-breeding and breeding. And this is one of our first results. In the last 2 years, we already improved the first parents of banana, which carried resistance to TR4, as you can see. Also, of course, they are susceptible. And additionally, some of them were already tested for Black Sigatoka, and some of them are resistant to Black Sigatoka. And interestingly, some of them are also resistant to both of them. So this is the initial, it's one more step in the development of a new variety. Now, to summarize, René already mentioned these very important pillars in our research towards the development of new varieties with resistance to these diseases. And if we talk about conventional breeding, it takes 15 to 20 years to develop new varieties. We have the same pillars, genetic resources, the crop knowledge, of course, the green fingers, and at the same time, the use of little technology. That's how breeding in banana has been done in the last 50 years. In Kijin, of course, we are trying to incorporate all those tools in order to make it fast, and we are convinced that with the implementation of all these different methodologies, the implementation of DNA, RNA technologies, we can start delivering new varieties in a time of 6 to 10 years. Finally, I want to acknowledge Kijin, of course, for the invitation to open this space for us, the banana team at Kijin, of course, our partners, and all of you for your attention.
Thank you. Thank you so much for your insights and for your information. So we are ready to take some questions from the audience, if there are any questions. I don't see any fingers. I don't see any questions. So your presentation was very clear. 100% complete. Thank you so much. Let's check with Eric if there are any questions from the online version of this event.
Not for Fernando and for René, but we do have questions for the other speakers.
Okay, so maybe we can invite we're running a little bit late, but why not? So if I can ask you to shuffle a little bit, and then I think we have a question for Dominic and we have a question for Guus. So please stay with us. Yeah. So Eric, we have a couple of questions coming in.
Yes, a question from Laura for Dominic.
For Dominic.
Dominic, do you already use molecular markers for screening on disease resistance, or predicting, I should say, disease resistances?
We start.
That's a very short answer. Yes, you're just beginning implementing this.
Yeah, but in general, yes, we start, and also our competition, I would say, they start. Yeah, I think we are in the same situation, what I described before, that we are lacking behind of the agriculture, but this is also our luck, I would say, because now we find this either major QTLs or qualitative traits for these diseases, if it's available. More problematic is, I would say, that the disease as itself, there is not so much knowledge about this disease. They are not described in such a proper way than in the agriculture. And to do then some phenotypic test with them, this is more problematic, I would say, than the afterwards analysis with genetic tools.
Yeah. Before we go to the question for Guus, Fernando, please step forward, because you're also one of our distinguished guests. So there's also, Eric, a question for Guus.
Yes, indeed. Thank you, Paul. Siuli is asking, it's about a bit of a diversion from DNA, but still it can be very important. Is the fully automated high-throughput phenotyping facility crucial when it comes to accelerating breeding in potatoes?
Yes, I believe in objective phenotyping, and I also believe in, if you speak about yield, that you try to find the components who together will determine the yield. On the other hand, we are an outdoors crop, and we are playing a lot already with indoors testing, with precision phenotyping. At the moment, we find the connection of what we find indoors with new technology, we will be the first to have this involved in our breeding program. But there are some complications because we have to do with a zinc source with potato, where the source is giving the potato the yield, and the relation between the leaves and the tubers is very important, and that is really a challenge to have that in quick and specified test indoors. But we are working on that as well. Yes.
Okay.
So yes.
Thank you for your answer, Guus. We also, Eric, I understand, have a question for Fernando.
Yes, indeed. Thank you, Paul. So there's a question about making choices in breeding when it comes to sustainability on the long term. Even when you breed a resistant Cavendish, so reconstruct it and make it resistant, isn't it risky to replace the existing variety with another monoculture, you could say? Do you plan to breed specific varieties for specific target environments? Or do you plan to breed for specific resistances for specific races of the Fusarium pathogen?
Yeah, it's actually a very good question. And indeed, I'm also convinced that the future is diversity. So we cannot continue with the same system in banana. I always say if we make a new Cavendish, probably we will have a new Fusarium tropical race 4 in a few years. So it has to change. But of course, the urgency is an important business for the industry. But at the same time, it's important for the local, because most of the Cavendish bananas are also produced in India, for example, in Brazil, for local consumption. So it's very important, and of course we have to fix that problem first. But at the same time, as breeders, you can never go in just one direction. You have multiple branches, and at the same time, we are also considering bananas with resistance, but with different color, for example, different taste that can be enjoyed for people in the future. Because especially in the market here in Europe, in the US, only one type of banana. But if you go to Southeast Asia, to Colombia, for example, you have broader options to other types of bananas. So definitely, Cavendish is the first objective, but at the same time, we are working on the generation of new diversity.
Yeah, and to put the emphasis on the importance of bananas, because it's the #1 fruit in the world, the most eaten fruit in the world, and also, as you mentioned, it's very important for the World Food Program, because a lot of the bananas are eaten for the internal market. Indeed.
Yeah. Additionally, we have also the cooking type of bananas, which are staple food in Africa and in America. So it's very important to make resistant varieties.
Interesting. So, any questions from the audience? I see 2 people having a question on the second row. Please stand up, sir. Yes.
Why is the banana Cavendish so dominant in the world? Because so many other varieties.
Yes, that's also a very interesting question, and it's the most common question always. Why and it's true. It's also a question for me. I would say genetically, Cavendish is just an amazing product. Yeah. So it's it's an herb that produces 30 kilos of food. Everything is in this monoculture system. Everything is fixed for the Cavendish size, time of harvesting, how to grow it. So I want to think that it's too difficult for the industry to change everything again, to introduce new varieties, but at the same time, the other varieties are not ready. So they also need to be bred, because they are either they have short life shelf, or they are still susceptible to other diseases. So
so commercially, the Cavendish is the most important banana. It's the easiest to grow, and it has the best quality.
Yeah, and it's a remarkable thing in Cavendish is that I think I cannot have another example similar to Cavendish for more than 100 years, still resistant to the race 1. So the same soils that were used for the Gros Michel are used now for the Cavendish and still resistant. So now the risk is the TR4, but if there is no TR4, they can just continue with Cavendish.
I have the feeling we can talk for hours about the banana.
I can, so
which is a very good thing, and maybe it's a good idea. Any more questions? I have one more questions in the back. That's the last question, I think, because we are running a little bit out of time.
Thank you. Question for Guus, a little bit of technical question. You showed that you implemented markers very successfully. You used a SNP platform for that, but you have a tetraploid crop. So you have to somehow deal with the ploidy and with the dosage. How did you do that? Because it's something that some other crops see as a threat. And also, is the dosage important for the expression of the traits?
That's a good one. Thank you. Andreas, indeed, it is important for your crossing strategy to know the frequency what you can predict for your in your offspring. Indeed, the most, for instance, resistant gene simplex is enough, but still we profit a lot to have at least the possibility to determine the allele frequency. So yes, and indeed, yeah, we don't like the fact that potato is tetraploid. Indeed, so but we have to deal with it. But we have also a diplet breeding program in which we can do much better studies on genetics and get rid of the interference of all those levels of frequency of the alleles. So yeah, that's one of the solutions to make speed and to get accuracy in the program.
So does that answer your question? Yes. Perfect. Thank you so much. So I'm afraid this is all we have time for today. Thank you very much for your contribution to this event, and also I would like to have a big hand for our panel members who are sitting there in the second row for their insights and their commentary on this really interesting event. So if I can have a round of applause, please. Thank you. And of course, last but not least, everybody here, thank you for attending this event, and also the people that are watching at home or from their workplace or their laboratory or wherever they are watching this event. Thank you so much. That's all for now here from Kijin in Wageningen. I hope you had a really interesting event and hope to see you next time. Thank you very much.